Okay, so today what your notes are going to be on is the scientific method. Now, you've already done the marshmallow challenge, and so I'm going to use the marshmallow challenge as kind of the guideline as we go through the scientific method. One of the things that often gets misunderstood when we talk about the scientific method is that we only use it in science, and really we use it all the time. It's basic problem solving, and you see it a lot in engineering. And you see it a lot in just general everyday life, and it's really a process. We have certain terms for the different sections of the scientific method, and that's more for convenience when we're reporting and when we're writing out lab reports than for there really being a very strong distinction between each part. So, getting started, the part that we always kind of have to start with is the idea of your research question, your problem. What is it that you are trying to figure out? So, for example, in the marshmallow challenge, it was who could make the tallest tower with the materials provided. Okay, that was the problem. That was the question you were trying to answer. So sometimes you'll see this referred to as the question, sometimes as the problem or a problem statement. It is generally written in a question format because it's what you're trying to figure out. So that's our research question or our problem. Our next piece is you come up with what you think the solution might be, your hypothesis. A hypothesis is not an educated guess. I really don't like that term because by saying educated guess, that stress, that connotation of the term guess is that you don't have any idea what really is going to happen and you're just kind of guessing at it. Usually when you run an experiment or you're doing something, you have a pretty good idea whether or not something is going to work. So for example, with your marshmallow challenge, that first thought you had, whether you decided to go with the TP, whether you decided to try and make a square or a cylinder type shape, how you decided to brace it, that was your hypothesis as to what structure was going to be the tallest. So that was your hypothesis. That was the your answer, your proposed answer that you went with. It is always written as a statement. A lot of times in science, what we do is we utilize the if-then statement. So if I do this, then this is going to happen. As you go through your high school career, the farther you go, you kind of start steering away from the if-then format. We really like it to start out with because it forces you to have a testable hypothesis. It's very hard to write an if-then statement and not have it be testable. However, when you get into advanced research, a lot of times the questions aren't quite simple enough to put in an if-then, and so you start steering away from those. We do base them on some prior knowledge. You don't just guess. So that's why I say it's more of a proposed answer. What do you think that actual answer is? It's less of a true guess and more as what you're proposing the accurate answer is. Now, once you have this idea, then you actually do the experiment. This is when you actually built your tower of spaghetti, string, and tape. And you went through, you planned it, you organized it, you try and do it under controlled conditions. The reason why you want to do it under controlled conditions is if everybody is trying everything all at once, then you don't know which part didn't work, whether it was the arrangement of the spaghetti or how you were taping something. And so by trying a couple of different methods, one at a time, you're able to then get the best method. Okay, and that's when we do our experiment, is we test out whether or not our answer was correct. And we test it out in, you can do multiple experiments for the same hypothesis because you're trying to figure out which one gives us the best answer. You always keep very careful records as you're doing the experiment so that you can't be accused of bias and so you can repeat it. If you can't repeat an experiment, you might as well not have done it. It's not considered valid until it gets repeated, and realistically not repeated once, but being able to be repeated multiple times by multiple different people and people who have no relation to you or even know you should be able to repeat your experiment for your hypothesis to be really considered truly supported or not supported. Now, one of the things that you'll see, we don't do this as much at the biology level, but as you move on, you will start seeing things like logbooks. Now, logbooks are basically a notebook used just for that particular research. When you have that research that takes maybe months or years, you put all of this data, this hypothesizing, all of the trials, everything you've done into that logbook so you can go back and figure out what you did, what you want to try next. Alright, so after you run your experiment, or as you're running your experiment, some of the things you need to keep in mind is you need to have your variables situated. So you're going to have a lot of things in your experiment that don't change. For the marshmallow challenge, everybody got the same tape. Everybody got the same string. Everybody got the same type of spaghetti and amount of spaghetti. Those were all constants. They were all controlled variables. They were things that were not changing. And also our dependent variable, how we were measuring it, that we were measuring it by height from the marshmallow down. These are all things that just are not changing how we're measuring or what's going on with them. Then you have one element that will change, and that is the element that we call the manipulated or independent variable. It's the one the investigator, whoever's doing it, is trying to change. So if, for example, you first tried a tripod structure for your tower and it didn't work, and then you went with the square cube type of tower, the shape of the tower would have been your manipulated variables. If you went from, instead of doing that, you said, okay, no, I like the tripod shape, but I'm going to try taping it in a different way, then you changed your independent variable there. So that's what that is. Now, controls of a sample. We have our controlled variables. These are just variables that we are keeping the same. So it's the same type of spaghetti, the same type of tape, so that you can't say, oh, well, I didn't do as well as this group because of fill in the blank with whatever excuse. If it's truly controlled, there aren't any excuses. You've covered them all. Everything has been controlled. You also have a control group, okay, and that is considered the control, capital T. The control is a sample that's treated exactly the same as everybody else, except they aren't exposed to whatever the variable was. So, for example, in the marshmallow challenge, if you were changing how you were taping things and you left the first way that you taped it exactly the same, well, you can then compare all of the other methods you were using to tape back to that first method so that you can actually see whether or not it was working better or worse. That was our control group. It's what you compare to. Okay, so those are our variables. The next thing you want to make sure you have in your experiment is a list of materials. And you don't want to just say a beaker. You want to actually be clear. Say it's a 250 milliliter glass beaker. That's important because if someone's trying to recreate your experiment, they need to do it exactly, which means using a plastic beaker versus a glass beaker might not make a difference, but it might. And so that's a variable you want to control. Alright, you then write out your procedure. Now, when we write out procedures, we want it to be step by step. And it's how you actually conducted the experiment, not how you were planning on doing it. How did it actually happen? And it should be very, very explicit in its detail of how you did it. The best way I have of explaining this is, imagine you are trying to get a toddler to follow those instructions, and the amount of detail you'd have to go through and how much you'd have to break down your instructions. That's the level your procedure should be at. That way, someone who doesn't know you, doesn't know how you speak, who doesn't know how you write, has never done the experiment, has no background on it, can read through your procedures and do it exactly the same way as you did. Okay, so for example, if after 1 minute I stirred in the baking soda and timed the reaction, you need to say how long after. Realistically, you should also have how much baking soda. Were you timing it in seconds, minutes? What were you using to time? These are all little details that make a procedure a lot stronger as far as in your lab reports. So once you have your procedures and you've got your materials and you've got your variables all figured out, then you actually collect your data. You do the experiment. And as you go through and do this, you want to record all of your data, record any observations. Those are the things that we have with our 5 senses: eyes, ears, nose, mouth, touch. And you also record things that may or may not change during the time of experimentation. So if, for example, I'm looking at plant growth from light, I might also keep a record of, well, what's the temperature like outside? What's the humidity like? Because that might be a contributing factor to other differences that I might see. Once you have your data, okay, so for this, our collecting data, relating it back to our marshmallow challenge, this is when we actually had our towers built and you measured how tall did they get. Were they the winner or not? Now, this then goes into the results. So usually we make this as a nice statement. It explains the data. You don't have to go into any detail. You don't have to explain what you think it means. It is purely what is the data in words. So when you collected your data, it should have been in a data table. It's not always super pretty. When you go through and you state your results, this is when you sum up. You have done your averages. You have figured out exactly what the measurements are and what the differences are, and so you can just simply make a statement of, this is what happened, this is how it happened. So for example, in the Marshmallow Challenge, once you've gone through and had all of those towers measured of how tall, you then had the statement of, well, group 5 won because they were X number of centimeters tall for their tower. Okay, now a lot of times when we get to our results, if the results weren't something that made sense or they weren't ones that we really like, then a lot of times what we do is we loop back up to where the hypothesis is, because remember, it's a process. So if when you were going through and you made your tower and you measured your tower, even if it wasn't the official measurement, you would see, my tower doesn't look quite as tall as the group over theirs. Let's go back and loop back up to the hypothesis part and fix it. Try something else. Try a different proposed answer. Okay, and so we go through that loop multiple times, trying to refine the procedures, trying to refine what it is that we want to do until we get a really good data set. Now, not with the Marshmallow Challenge, but when we do labs, what we do once we have those results and we're happy with the results and we've done several cycles of editing as far as our procedures and how we are testing this, then we want to actually show our results. And this is what we refer to as the analysis. Okay, it's our charts, it's our graphs, it's in words. You illustrate basically what is the data happening. What does it mean? So when we do line and bar graphs, it really makes it, the relationships a lot clearer, and it makes it a lot easier to tell what happened in the actual experiment. We also see this sometimes in just simply words. When we say, okay, well, the more light that tomato plants get, the taller that they grow. Or the more light that the hosta get, the less well they grow, because hostas are a shade plant. So as we go through and once we have our analysis done, then we can make a conclusion. So this is a basic summary of all of the research. So we go all the way back up to our problem and we sum up, well, this is what our problem was, this is what our proposed answer was with our hypothesis, this is what we did, this is what we found out, and this is what we think is kind of going on. You do want to make a statement of whether or not your hypothesis was supported or not. You may have data that didn't support your hypothesis, and that is perfectly valid. Okay, now going through, our last piece here is kind of the flow chart here. We start with ask a question, do a little background research, have a hypothesis, test it, analyze it. If it's true, we report the results. If it's false, we report the results and then think about it and try again. Our hypothesis should be